All right, we're going to kick it off, not to delay matters any further. Good morning to everyone. Welcome to the final SAEON Seminar for 2022. My name is Mfundo. I'm a scientist working with the Elwandle Coastal Node based in Agulhas, and I'll be your host for this morning. This is just a reminder that this session will be recorded and eventually posted on the SAEON YouTube channel. Please feel free to go view the YouTube channel at your leisure. All of our past presentations are on there, and it makes for good viewing. Just some housekeeping, please. While the presentation is underway, please keep your microphones muted. You are welcome to type questions in the comments box. We will view those after the presentation. After the presentation, we'll allow direct questions, which I will mediate. So with that, let me introduce our speaker this morning. It's my pleasure to introduce Dr. Mfundo Bizani. Mfundo has been part of the SAEON family for well over 10 years now. We first met Mfundo as an honor student for a very industrious project at the time that involved a 24-hour sample at sea, studying fine-scale zooplankton variability at that time in Algoa Bay. So after his honors, Mfundo decided to pursue a terrestrial feeding ecology project in the Fish River Valley, chasing yellow mongoose scat, of all things. Upon completing his masters, he decided it's time for fresh coastal ocean air again. So he joined the SAEON team first on an internship, followed by pursuit of a PhD project where he studied the plankton, zooplankton dynamics in Algoa Bay. Graduated earlier this year from that project. Very proud of Mfundo's achievements through that time, and it was a very demanding project. I think you logged over 70 cruises in Algoa Bay for the project, well over 100 now. In his professional capacity, Mfundo is currently appointed as an early career scientist at Elwandle Node in the portfolio of plankton ecology. So with that, Mfundo, I'm handing over to you.
Thank you, Sean, for that introduction. Good morning, everyone. Yeah. So just a little bit of introduction. Why zooplankton? Often I get asked by high school learners that, okay, why zooplankton? And the first thing that comes out to me is that this is the most diverse group of animals. When we were at university, we were told no insects are the most diverse. But when I picked up a bookshelf book, that was not the case. And secondly, the most funny thing that I say to them, listen, guys, when you eat a hamburger or when you are eating a nice steak, you don't think about the nutrients of this on the soil. You don't think about the grass. You don't think about the actual cow. You're thinking about the burger. So zooplankton are a quite key component of the pelagic ecosystem, because they are the key component that transport or transfer the energy from phytoplankton to the hydrotrophic levels, such as fish. So this is why this is quite important to study zooplankton. And then secondly, why Algoa Bay? That we will get into a little bit shorter, a little bit later. Okay, so this is the outline of my talk today. I'm going to talk a little bit about Algoa Bay and give an overview about the pelagic ecosystem that's run by SAEON, and then give you some of the results on spatio-temporal variability of zooplankton biomass, which is some sort of measure of zooplankton standing stock. So this data will cover from 2012 to 2016. So this was part of my PhD, and then give a little bit of overview of what drives the variability that we see in zooplankton biomass, and then I will end that section with highlighting a unique feature that we've identified in the bay, and then give a very short comments on temporal trends that we've been observing since 2010 until 2016, and then highlight on a large-scale harmful algal bloom that was caused by a non-indigenous dinoflagellate, which highlights the changes in temperatures, which can be directly linked to climate changes, and then end off with some concluding remarks. I hope you enjoy. Okay, so Algoa Bay is quite unique in the sense that it is situated in a region where it is high influenced by the Agulhas Current, which is generally warm, salty, and fast flowing, and it's in the transition zone between the west and the east, and most probably that's the reason why some people say it's a windy city, because it's a quite dynamic environment. So the influence of the Agulhas Current in Algoa Bay is that sometimes there are natal pulses that passes through that causes an upwelling. So upwelling is a term where cold bottom water that are nutrient rich transported towards the surface so that they are available for phytoplankton that should transfer that to zooplankton and fish productivity. And it is quite unique in the sense that both wind and current-driven upwelling occurs in this region, particularly at this region, the eastern sector and the western sector. And it's interesting in that on the western sector, actually the upwelling occurs outside of the bay, and sometimes the water can be transported inside the bay. We will talk about that a little bit later. And this process is usually occurs most dominantly during the summer seasons when the easterly winds are more dominant. Okay, so every month, SAEON Elwandle Node goes out and sample at 8 different stations, marked by the crosses, and then it also has continuous temperature lockers that are moored towards the seafloor, and they collect temperature every hour. So these sites were not just randomly selected. They were selected because to try and understand how the different zones of the bay will have, or how the different zones in terms of oceanography, in terms of zooplankton and phytoplankton productivity will differ. So the first 2 stations I've highlighted here, the Cape Recife in the western sector and the Woody Cape in the eastern sector. Those are the upwelling hotspot. Please, we must keep in mind that the western sector, which is the Cape Recife side and the Woody Cape, those are the upwelling hotspot. And then these 2, station 1 and station 2, are situated in the leeward side of the harbor or of the bay. They are sheltered. Potentially, especially site 1, they have anthropogenic influence. And then station 3, which is situated just off Sundays River, is a semi-exposed station, which should potentially also have influence from the Sundays River, which has a heavy farming activity. And then station 4, which is just offshore of Alexandria Jewel Field. This is a plankton hotspot for the surf zone, and potentially there should be enough or sufficient fresh water supplied from the dune fields that should ultimately support a high primary production and secondary production. And then these two stations, station 7 being the only one that is situated at a 60-meter contour, and the rest of the stations are at the 30-meter contour. We anticipated that this should be highly influenced by the Agulhas Current. So trying to capture all of that so that we can see the bigger picture of what the ocean dynamics are doing in Algoa Bay and relate this to productivity. So in the past we used to use Kwabelana, but now we've got other boats like Kalanas and Observer. So every month, like I mentioned, we go out, collect physical chemical parameters using the CTD, figure C there. We collect temperature, salinity, turbidity, dissolved oxygen, and on top of that, we collect water samples for analysis of nutrients, such as nitrates, nitrites, ammonium, phosphates, and silicates. And then this is the WP2 net fitted with a flow meter to record that is used to calculate the amount of water that is filtered when we're collecting zooplankton samples. And this is a typical thermistor strings that is deployed towards the seafloor to continuously monitor temperature. Okay, so just to give a little bit of results of what we have seen in terms of temporally and spatially in zooplankton biomass since from 2012 to 2016. What we've seen temporally was quite interesting in all of the stations from station 1 to station 7. Mind you, station 8 is not included here because it was sampled a little bit later. So we will talk about that a little bit later as well. So what was interesting to see in all of the station was that towards spring, there seems like zooplankton is increasing from spring to summer, and then autumn to winter starts to decrease. And what was interesting to notice was that this from spring to summer, this is the time when upwelling starts to occur more frequently in Algoa Bay. So we linked this to that at first, and then when we now looked at environmental variables, this was a mission for me to convince my PhD promoter, was that all of the variables that we looked at were significant predictors of zooplankton dynamics, and except for ammonium and chlorophyll. And this was strange. So this was when we performed generalized additive models, and this was strange to find out. Okay, why chlorophyll is not a significant predictor? Because this is a proxy of phytoplankton, and we see things like turbidity and some nutrients being significant predictors of zooplankton biomass. My explanation for this was that it is some sort of temporal mismatch. If you feed on something for you to increase in number, that means you are taking up a lot of phytoplankton. So that was some sort of explanation that I tried to convince my PhD promoter regarding this section. So what was also interesting was that when we looked at things like silicates, they appeared that zooplankton tend to increase when silicates increase. And in terms of what this means is that because things like coccolithophores and diatoms are silicate-based cells, this is some indication that they were in the water, but they were used up by zooplankton. And if you think about it like this, Diatoms are the most nutritious phytoplankton group for zooplankton. And then what was interesting for me as well was that when we compared stations, when we tried to test if there is any significant difference in zooplankton biomass between stations, this was not the case. We couldn't find this, and it was very frustrating for me. But what we were able to identify was that the two stations that are located on the western sector, particularly the ones in the sheltered region, generally had higher zooplankton biomass compared to anything else. And we were not surprised to see that Station 7 has had lower zooplankton biomass, because this is an offshore station that is situated in deeper waters, and theory suggests that the more you move away offshore, you tend to have less biomass but high species diversity. Secondly, what was interesting, or what the generalized additive models were able to confirm for us quite clearly, was that both spring and summer tend to have high zooplankton biomass, and these are the times when upwelling occurs more frequently in the bay. And we were not surprised to see this because in Algoa Bay, during the winter seasons, westerly winds tends to dominate, and the water column becomes mixed. And for phytoplankton to be able to utilize the nutrients, the nutrients to be towards the surface, and a little bit of stratification needs to occur, stratification or relaxation. And then when we were trying to pin down what was the most important parameter that was a significant predictor for zooplankton biomass, when we were looking at just the environmental data from 2012 to 2016, you can clearly see from this PCA that there is no pattern in terms of environmental drivers. And this was quite shocking for me, because we expected to see both summer and spring isolating, and winter and spring, winter and autumn being grouped together. So this was quite frustrating, and when we dig a little bit deeper, we find that, okay, the cause of this is the ocean dynamics of Algoa Bay are so variable. Unlike the West Coast, upwelling does not occur seasonally. It occurs at any random time when the easterly winds occur. So what we were able to identify was that, firstly, if you look at figure 8 here, you can clearly see that there is a difference in the water temperatures on the on the west side of the black arrow and the red arrow. And 4 days later, you can see that upwelling tongue that was starting to occur on the 22nd of January has actually expanded westward towards the entire bay. So that is most probably one of the reasons, because maybe we went sampling this day. Maybe we went sampling this day because we go sampling opportunistically. And then if you look at this upwelling, that event that was happening on the 3rd of March, in March 2010, clearly you can see that at both capes, upwelling was starting to occur. And this, of course, the Cape Recife site was more prominent than the Woody Cape Padron one. And a day later, you can see there is clearly a warm water that is sitting in the western sector of the bay. That means that there's nothing that has been advected offshore. So this region means that the that warm water that was sitting there has been actually advected offshore. And then a day later, again, you can see there's a warming that is happening on that side. So this is adopted from Goshen's 2015 paper. And then again, you can see that, okay, there is an upwelling that was happening on the 18th of April. And then that upwelling never even entered the bay. So this is some sort of, so which means the variability occurs irregularly, and it can occur anytime. So that is why it is very difficult for a project like ours to be able to capture specific to distinguish between the sites in terms of environmental variables. And this is why this has transpired into zooplankton biomass. The difference in Algoa Bay and what we were the unique feature that we said we have identified. So this is a paper published recently, but the upwelling shadow was first identified, I think, by Graham back in 2008 or something like that, if I'm not mistaken. So unlike in Algoa Bay, in Monterey Bay, the upwelling only occurs on the northern side of the bay, and that water sips down and gets transported into the bay, but it never reaches the coast at this side of the coast, and that is a permanent feature in that bay. So that is why they can easily say, okay, this site and this site is completely different. Unlike in Algoa Bay, if you remember, upwelling occurs on this side or in this side, and if the westerly winds blow, for a prolonged period of time, that upwelling tank that occurs here covers the entire bay. So this is why it is completely different, and that feature then tends to be here, it can be here, it can be there. So that feature tends to move around. It is not a permanent feature, so it is not located in one region. So what is important about this section of the bay? This shadow has been identified to be a plankton hotspot because there is cold water that cold nutrient-rich water that comes into the bay, and then this warm water that creates some sort of horizontal front where the conditions are very conducive for both phytoplankton and zooplankton, because we have enough sufficient nutrient supply, and the water is not extremely cold, and the currents are a little bit are not as strong, and so the force of advection does not affect them. So this region has been but specifically identified as a plankton hotspot. So clearly this is a typical sampling that we did in Algoa Bay, and we could identify exactly the same phenomena using our in-situ data. So you could see this figure A is the SST sea surface temperature from the CTD. You can clearly see on the eastern sector of the bay there was an upwelling event that had occurred, and you could see in that region but there was no upwelling that had occurred in the leeward side of the harbor, and that has transpired into that there was high zooplankton biomass. And this can mean one of two things. It is probably upwelling shadow, or whatever that was here initially before the upwelling of the event had occurred. That plankton had been advected offshore. So which means that upwelling has two things. It can be detrimental or it can be conducive because it brings up cold nutrient-rich water towards the photic zone, and then phytoplankton can use that. But in this instant, it means that it has been detrimental because whatever one that was there, if that is the case, had been lost offshore. And then I'm going to talk about a little bit I'm not going to go into detail in this one about temporal trends that we have been seeing from July 2010 to April 2016. So when we performed time series analysis, we could see clearly from 2010 zooplankton biomass has been increasing in the bay, and I want you to pay attention in this arrow and this the red arrows, and this the most significant increase that we have seen occurred late in 2013. And this has coincided with an event that I'm going to explain later. So when we performed structural breakpoint analysis, we could see that almost all of the stations had increased in a similar fashion and almost in synchrony. And why is this important to understand that? So there was a theory that was developed initially developed for pelagic fish to understand what would be the influence of upwelling as on fish stock, and this has been now adopted to explain also temporal variability in zooplankton dynamics. So when like I explained, upwelling tends to support primary and secondary productivity, right? So what we want to see is that when nutrients are being supplied towards the euphotic zone, there will be an increase, but there is a certain range that that will cause actually a loss of zooplankton as climate changes. When the upwelling favorable winds tend to increase as well, and like I explained on the other graph, that it tends to advect offshore whatever was towards the coast. So this is quite important to monitor. But at the moment, it appears that in Algoa Bay we are still in a weak, low to weak turbulence or advection. So maybe we're still good. It is, but we still remain to see if this is going to apply. This dome shape is going to apply in Algoa Bay, or this is just a decadal trend or something like that. And then I am going to, this is my last section. I'm not sure what I'm doing with time. So here I am going to highlight the impact of climate change and what it is doing to the zooplankton of Algoa Bay. So this is an episodic event that occurred around in December of 2013, when we saw that huge spike in zooplankton biomass. So this was caused by a dinoflagellate that does not originate in South Africa. We don't know where it comes from, but literature suggests that cysts have been recorded in sediment samples, and this cyst can stay in the sediment for up to 50 years, even until the conditions are favorable for them to actually bloom. So we've never seen this species before. It is potentially toxic, depending on the strain, because it can cause it can produce the toxins. So this is was quite important to highlight. And this, without, I like to emphasize this, without the continuous monitoring of that SAEON undertakes, we were never going to be able to pick up this event. So firstly, just to highlight the role of zooplankton in harmful algal blooms. Harmful algal blooms are generally referred to as red tides. So they can sometimes, so zooplankton are selective feeders. They can detect and say, no, this is not what I like to eat, and avoid it. So because they will can select, they can choose to feed on other phytoplankton species, leaving the one that is potentially toxic and can cause red tide to actually bloom. So by not grazing on them, and they can be suppressors, because they can actually, depending on what species it is, they can actually graze and suppress the bloom of the harmful algal bloom, or they can have no impact at all, because sometimes these dinoflagellates produce toxins that actually affect their productivity, their feeding, or even paralyze them. So that is the main general role of zooplankton in harmful algal blooms. So in Algoa Bay, harmful algal blooms generally occur naturally, and they are commonly caused by noctilucans sentinels, which is quite a cosmopolitan species. This species is quite large. It thrives because it was one of the fascinating dinoflagellate, because it can reproduce, one, sexual and asexually, and two, it is mixotrophic. So it can have a symbiotic relationship with the cyanobacteria that can photosynthesize, or it can feed on diatoms, on copypods even. So this is why it is quite an interesting one. And yes, it causes water discoloration, so red tides as well. So this is why this species is quite a cosmopolitan one. And it's quite important also to start it, because it can actually fit on copypods as well. So this bloom that was caused by Lingulidinium polyedra was the most extensive one that we've ever seen in this part of the world. It occurred approximately about 4 months and covered quite a big spatial from Mossel Bay up to almost East London. It caused there were fish kills, because when they decompose, they tend to when they decompose, they tend to the reaction that happens tends to deplete the oxygen towards the seafloor, and then this causes fish kills. It is potentially toxic. So whatever, if things like mussels feed on them and we eat those mussels, and we can have those toxins as well. So they can serve as vectors of those toxins as well. So just to give a background of what caused this this bloom to occur, as I mentioned, this was the first time we've ever recorded this bloom. So typically highlighted here are the easterly winds that causes upwelling in Algoa Bay. They occurred for more than 7 days. Usually these winds in this region occur maybe 2, max 3 days. So this one occurred for approximately a week. And if you can see in the 3 stations here, Cape Recife, Woody Cape, and St. Croix, there was a huge event, a prolonged upwelling event that occurred. That was followed by a warming event. You can see at 30 meters, water sitting at around 22 degrees Celsius. That occurred, and then the bloom started to occur late December when there was that prolonged upwelling, and then the sea were calm and the conditions were quite conducive for Lingulidinium to Polyedra to bloom. And you could see these are the fluorescence readings from the CTT. You could see that in December 2013, when we went sampling, fluorescence reading where the maximum was up to 8, and then January, February, they spiked up to from 50 to up to 500. What was quite interesting for me as well was that these are ordered from Station 1 in the western sector, and then this is Cape Recife. What was interesting for me was that to see that, again, the western sector of the bay tends to have high productivity, and which means there is some retentive capacity that is happening in the leeward side of the bay. And then when we looked at the long-term mean from 2010 to 2013, just before the bloom, we could see that, of course, zooplankton biomass in the bottom right figure was significantly increased. This coincided with what we've seen in the temporal series chapter. We could see chlorophyll A spiked from January, February, when the bloom was at its peak. And what was interesting for me was to look at silicates, because it appears that things like nitrates, although this has happened after an upwelling event, well, that was quite extensive upwelling event, that nitrates were generally decreased, which means that though that could be the one of the limiting factor for Lingulidinium Polyedra, whereas things like silicates, it does not prefer to use them. And when we looked at species composition from the period of June 2013 to June 2014, with the red box highlighting the period where the bloom had occurred, we could see that the first thing that we noticed was that there was a spike of tintinnids, which are generally microzooplankton, and we have never seen this before the bloom had occurred. We only seen them during when there was that bloom, and it appears that they tend to be in regions where they tend to be to dominate in regions where dinoflagellates dinoflagellate blooms tends to occur. And what was interesting is that also this species can form cysts as well that can sit on sediments for quite a long time, and when conditions become favorable for them, they can also bloom. And this is not good for the pelagic ecosystem, because you want to have higher calanoid copypods, the bigger ones that fish larvae fits on. So an environment that is good should be dominated by medium to large calanoid copypods that then support fish larvae. But what we've seen in that period, there was a spike in increase in smaller ones, smaller cyclopods, your orthoina, gelatinous zooplankton on the bottom ones, catocerans and doiliolids. And even the gelatinous zooplankton are not nutritionous. So they don't provide nutritious food for fish. So you don't want to see them bloom as well. And then when we did a little bit of statistical analysis, we could see that the period before and after the bloom, so the green and the black dots tended to have a similar zooplankton composition as opposed to the red tide period. And this was primarily caused by tintinids, polychaete larvae, Plutus larvae. We're not sure if the Plutus larvae would have been increased because of the bloom, or was it that they were dying off because of the toxins or the depletion of oxygen. But clearly, these three groups tended to increase. In closing, we say that, okay, Algoa Bay is quite a unique dynamic environment, which has upwelling that occurs irregularly. We have identified Western sector as a region of generally high zooplankton biomass because of its retentive capacity. The most prominent variability that we've seen in zooplankton was temporally or seasonally. And upwelling appears to be a key driver of that pattern. And we've identified a significant increase in zooplankton over time. And we say the bloom of this nature that had occurred in summer 2013-2014 is more likely to occur because we have seen similar ocean conditions like that. And what we have recently noticed that salps have been blooming in Algoa Bay, and we haven't observed. This is actually a sample that we collected in Algoa Bay, and we haven't seen this before the period of 2013. With that, I'd like to thank you all for your time and for listening to me. Thank you.
Thank you very much, Mfundo. Very interesting talk. Lots of work done. Thank you very, very much. I'm going to open the floor for questions. I did not note any questions coming through the comments, the chat box. Yeah, that's correct. So the floor's open. Any questions? Okay, I'll start with a question, Mfundo. So yes. I cannot believe it's almost been 10 years since that pivotal event. You know, it's clearly had impacts on the bay. We're still trying to figure out exactly what the consequences are for the ecosystem in the long term. So my question is, what is next? Where does your interest take you next in terms of new projects? What do you intend to pursue now?
So there are a couple of questions that had came up from my PhD, and one of the key things that I would like to look at is to understand if the strain of Lingulidinium polyedra is toxic or not, because we get different strains of these dinoflagellates. So to see if the one we have is toxic, and how if it is or if it is not, to key copypod species such as Eucalanus saccalensis fit on them or not. If we subject them into monoculture or culture with diatoms, if they will select Lingulidinium or most dominant diatom species. So that is one of the key things that I would like to look at. And the second thing that I want to look at is what is the cause of that increasing trend in zooplankton biomass? Is it linked to a particular species? Is it linked to, say, salps? What is it linked to? So is to really get an understanding in terms of species composition. What is driving that? So that is a second thing I like to pursue.
Very interesting stuff. Thanks, Mfundo. I'm going to try again. Any questions from our audience? I think that is it, Mfundo. Thank you very much. We really appreciate your presentation. Enjoyed it tremendously. This is just an announcement that the next SAEON talk will occur on the 3rd of February in the new year. We do not have a title yet, but we'll announce soon in the new year. So please look out for further communication in this regard. Then I'm going to wish you well on the holiday that is approaching. Good luck with the final few things that needs to be achieved before we break for holiday. Take care, everyone, and see you in the new year. Thank you, Mfundo.
Thanks, everyone. Thanks, Sean. Thanks, Caitlin.
Thank you, Caitlin. Bye-bye, everyone.